The overall goal of this noxious cold assay is to be able to quantitate and assess changes in innocuous to noxious cold detection.
This method can help answer key questions in the nociception field, including facilitating identification of novel, conserved cold nociception genes and modulators of pain responses in disease and/or injury.
The main advantage of this technique is it allows for precise manipulation of a noxious cold stimulus and quantification of cold-evoked responses over a variety of environmental and genetic contexts. Generally, individuals new to this method will need to practice consistent application of the probe before beginning an experiment.
We first had the idea for this method when we found that Drosophila larvae respond to noxious heat, and we became curious about whether they do the same to noxious cold.
Raise stocks or genetic crosses of Drosophila in a 25 degrees Celsius incubator. Note, if culturing a cross, use 20 to 25 virgin females and 15 to 20 males per vial containing regular cornmeal fly media and culture for approximately 48 hours before transferring flies to a new vial of food. 5 days after the culture is started, collect third instar larvae of the desired genotype by gently squirting a stream of water into the mushy food and larvae. Then, pour out the contents into a medium-sized clean petri dish. Next, gently sort larvae by size using forceps to preclude larger wandering third instar or small sickly larvae from being tested. Discard larvae containing any undesired genetic markers or balancers. Have another lab member label containers where sorted larvae will be placed to keep